Hi everyone, welcome back. Miss Calabrese here. In this video, we're going to be talking about the basics of your immune system. Right, so your overall immune response is commonly divided into two basic categories, and those are the innate response, which is inborn. That means that you're born with all of your innate immunity. And the other category is the adaptive response, sometimes called the acquired response, and that one you have to develop over time. And then we can further divide that adaptive response into either cell-mediated immunity or antibody-mediated immunity, and we'll talk about what those are in a minute. All right, so we're going to start by discussing the innate response. Right, so the innate response is the one that you're born with. Usually we will subdivide this innate response into a few different categories, and these are basically barriers to entry. So these are things that prevent pathogens from getting into your body, and sometimes I'll refer to this part of your innate immunity as your first line of defense. So these are external barriers to entry. So these barriers can be mechanical or physical barriers that literally stop things from getting in. They can be chemical barriers or they can be reflex barriers, right? So examples of mechanical or physical barriers would be things like your skin. So skin is a really good physical barrier that's going to prevent pathogens from entering your body. Your mucous membranes are also going to be good physical barriers that prevent things from getting in your body, things like nose hairs and eyelashes and stuff like that, that just kind of acts as a protective barrier. Also, even things like your normal microbiome. So your normal microbiome is considered a first line of defense. Those are microbes that are not pathogenic, usually, that live uh, on your skin or inside your digestive tract, and that those are going to be helping to defend you against pathogenic microbes. As far as chemical barriers go, those would be things like the acid in your stomach, right? So when you eat food that contains pathogens, a lot of those pathogens are going to be immediately destroyed by acids in your stomach, so very low pH there. You also have the pH of your skin is also a little bit on the low side, so slightly acidic. That helps to prevent the growth of bacteria. And then things like your saliva and your tears contain an enzyme called lysozyme, and that lysozyme is going to be useful for breaking down the cell walls of bacteria. Right, so those are all chemical barriers. And then reflex barriers are things like sneezing and coughing they're basically an ejection response. So something in your nasal cavity tickles you, you're going to sneeze to try to eject it. Something in your trachea, you'll cough to try to eject it. Even vomiting and defecation can be considered reflex barriers if they're trying to get rid of something that's entering your body. So here's some of those mechanical barriers again, and the chemical barriers. One other thing that I didn't mention as far as mechanical barriers go is this mechanical flushing. So the constant movement of fluids in your body is going to be an important protection against the buildup of pathogens. So the fact that you're constantly washing tears over the surface of your eyes helps prevent microbial growth on the surface of your eyes. You're constantly moving mucus around in your respiratory and digestive tracts. That helps prevent the growth of microbes. And then even frequent urination helps to flush microbes out of the bladder, right? So just constantly keeping things in motion is an important part of your innate immunity. Okay, and then these reflex barriers again. So anything that's kind of like a reflexive ejection of something in your body is probably a reflex barrier that's helping to protect you against microbes. Right. So all of those things, the chemical, the reflex, the physical, those were all first line of defense, so external barriers to entry. Now I want to move on to the second lines of defense. Second lines of defense are internal innate barriers. So we're still talking about the innate immune system, or the one that you're born with, but these ones are internal. So if a pathogen happens to get past that first line of defense and finds a way into the body, then we have second lines of defense, or internal innate barriers that are ready to protect us from that pathogen. So there are 6 different components of your second line of defense, and that's phagocytosis, which is just white blood cells eating pathogens, inflammation, fever, complement system, interferons, and natural killer cells. So we're going to talk about each one of these individually. All right, so first one was phagocytosis, and this one is incredibly important. So phagocytosis is something that a lot of your neutrophils and macrophages are capable of. So this is when they detect a microbe, a pathogen, and eat it, essentially. So they bring it in through endocytosis. Then once it's brought into the cell through endocytosis, it's then contained in a structure called a phagosome. The phagosome will then be combined with a lysosome, and that will cause the digestive enzymes in the lysosome to be released into the contents of the phagosome, which is going to cause that microbe to be destroyed. So that's mostly how phagocytosis works in neutrophils and macrophages. Inflammation is another part of your innate immune response. So we often think of inflammation as, something uncomfortable, kind of like a pesky reaction that our bodies have, but it is important to protecting you. So what's happening in inflammation is a lot. So there's a whole bunch of events that are happening that promote an inflammatory response. Now, the main characteristic signs of inflammation are redness, heat, swelling, and pain. So those are the 4 things that are kind of signs of inflammation. And so what's causing those things is a whole number of things going on in your body. One, we're going to be triggering if there's been damage done. So, and then, a damage to the outside tissue where we're bringing possible pathogens in. We want to increase blood flow to that area. And we're going to increase blood flow to the area because we want to draw in white blood cells that are capable of protecting us. So we want to increase blood flow to the area. An increase in blood flow is what causes redness. So more blood in the area, that tissue is going to look a little bit more red. Also, the capillaries in that area are going to become a little bit more leaky. So remember that some white blood cells are capable of diapedesis, meaning that they can leak out of the capillaries and into the surrounding tissues. So that increased permeability of the capillaries is what's going to get some white blood cells into the tissue, but it's also going to lead to swelling because as we lose white blood cells into the tissue, we're also going to lose plasma into the tissue, and that's how we get swelling. The swelling, but from the increased leakiness of the capillaries, is what puts pressure on surrounding nerves, and that's what creates the pain. Is just the swelling. And then the heat is a response to the increased blood flow to the area, right? So that increased blood flow brings more warm blood, which is heat. So those 4 things are caused by all of those different pathways happening to try to protect you. So inflammatory response is there to protect you, help you, but it can be uncomfortable. All right, another part of your innate immunity is a fever. So fever is when you increase the set point temperature of your body. So normally your hypothalamus is in control of maintaining regular body temperature, and it likes to keep it around 98.6 degrees, maybe a little bit lower than that now. So that hypothalamus normally likes to keep that set point, but you can trigger your hypothalamus to increase the set point if you have a whole lot of phagocytic activity happening, right? So a whole bunch of phagocytosis going on is going to mean that those phagocytes release a chemical called pyrogen. Pyrogen will then find its way to the hypothalamus, and the hypothalamus will take that as a note to increase the set point, so give your body a fever. And giving your body a fever, again, not always a comfortable thing, but it is designed to help you. And there are a couple different ways that a fever helps you. One is that it's going to increase the metabolic rate all over your body. So increase the temperature, we're going to increase the rate of reactions, which means we're going to be able to make white blood cells a whole lot more quickly to defend us from this pathogen. Also, some microbes just don't like that higher temperature, so they don't work as well, but you are producing more white blood cells more quickly. All right, so that's what a fever is for. All right, another part of your innate immunity and part of your second line of defense here is interferon. So interferon is a compound that gets made by cells that have been infected by viruses, right? So viruses need to enter into a cell in order to reproduce themselves. So they need to get inside a host cell, one of your cells, and then take over that cell. So this is something that once a cell has been invaded by a virus, it really can't do that much to protect itself. One of the things it can do is warn its neighbors, and that's where interferon comes into play. So interferon is a chemical created by a virally infected cell to warn neighboring cells to kind of shut their doors, shut and lock the doors because there's viruses in the neighborhood. We don't want to let those viruses into these neighboring cells. So interferon is basically a warning system to protect neighboring cells that's created by a cell that's already been infected. All right. Complement system is also part of your second line of defense. So complement system is a series of blood proteins. So these are proteins just dissolved in the blood floating around your body. They are a lot of times referred to as defense proteins because when they're directed to do so, they can basically poke holes in a bacteria and destroy it. But they need to be told what to do by antibodies usually in order to get this to happen. But complement proteins are proteins that just float around in the blood and when directed to do so, will poke holes in a pathogen. All right, and then natural killer cells. So natural killer cells are structurally similar to lymphocytes, but they are non-specific immune cells that are able to induce apoptosis in a compromised cell. So apoptosis is a word for programmed cell death. So basically tells cells to kill themselves if they've got an infection from a virus. So once a cell has already been infected by a virus, it's not able to be saved. A natural killer cell will go around and induce apoptosis by using some proteins called perforins and granzymes. The perforins pop holes in the membranes. Granzymes induce the death of a cell from inside. So this is going to find cells that have been infected by viruses, and it can also find cells that are cancerous. So when a cell turns cancerous, your natural killer cells are the ones that are going to be usually the first to arrive there and destroy that cancerous cell before it starts to spread. Okay, so difference between innate and adaptive immunity. Which one is present at birth and which one do you learn over time? Which one is non-specific and which one is specific? Right, so there's your answer. So your innate immune system is present at birth. The adaptive immune system you have to develop over time. The innate immune system is non-specific, so it's not recognizing the difference between specific pathogens, but it can tell what's a foreign entity and what is the body. And then the adaptive immune system is the one that is antigen or pathogen specific, meaning it can tell the difference between individual pathogens. All right. What are your second lines of defense? What are the symptoms of inflammation and how do you get a fever? Right, your second lines of defense are phagocytosis, inflammation, fever, complement, interferon, and natural killer cells. The signs of inflammation are redness, heat, swelling, and pain. And you get a fever by the release of pyrogens from phagocytic cells that trigger the hypothalamus to raise the set point of your body temperature. Right. So that was all second line of defense and first line of defense, which is falls under the umbrella of innate immunity. Now we're going to move on to adaptive immunity, which is much more complex and much more specific. So adaptive immunity it basically characterized by these two things: specificity and memory. So specificity meaning we're recognizing individual pathogens now, and memory meaning that if we've encountered this pathogen before, then we're going to remember it later. Right? And all of this specificity and memory is based on recognition of antigens. So antigens are foreign molecules or any molecule that the immune system is capable of recognizing as being foreign. So the ability to recognize antigens is going to be incredibly important in your adaptive immune response. All right, so again, antigens, any foreign molecule that can be recognized by your immune system. These foreign molecules, a lot of times, are proteins, but they can be smaller than proteins as well. And even individual antigens can have different regions on them that are able to be recognized by your immune system. Right, so those different regions of an antigen that can be recognized independently are called epitopes. So an epitope is just part of an antigen that's going to trigger its own immune response. So that means that you can, an individual antigen, so like this drawing here, say this whole antigen is a bacterium, that one single bacterium can have multiple epitopes or antigenic determinants, meaning that we can mount several different waves of response against that particular antigen because we can mount an entire immune response for each individual epitope. Okay, all right, so what makes a good antigen and what makes a bad antigen? So antigens, again, anything that your immune system recognizes as being foreign, but what it recognizes is going to have to do with the size of the foreign molecule and the structure of the foreign molecule. So one, it should be foreign. For the most part, your immune system should not be recognizing self proteins as being antigens. Usually, antigens are going to be organic molecules because organic molecules are a little bit bigger. So larger molecules are easier for your immune system to find. So that means things like proteins make really good antigens. And they also tend to be structurally complex. So really simple, repetitive structures like sugars or carbohydrates don't usually trigger your adaptive immune system just because of the simplicity of the molecule. So large, foreign, organic, complex molecules, those are the ones that act as the strongest antigens. All right. Another group of proteins that we should talk about is your major histocompatibility complex. So major histocompatibility complex is a set of genes that is going to be very important for the function of your immune system. Right, so these are genes that you have that code for proteins, right? Just like all genes code for proteins. The proteins that these MHC genes code for are your human leukocyte antigens. So these are, they're basically proteins that we find on the surface of your own body cells. Right, so we divide MHC genes into three different classes. And again, each class is important for immune function. So MHC class 1 genes. These code for proteins that we find on the surface of all your body cells, particularly all the cells that have nuclei. All right, so all nucleated body cells are going to have class 1 MHC proteins on their surface. And those are basically acting as ID cards, right? They're just self-identification proteins so that every single one of my cells, for example, has a little protein on the surface that basically says, I'm Alyssa's body cell, don't attack me. This is where I belong. This is the body that I belong in. So every one of your cells also has these, and these are very specific to the individual. So nobody else is going to have my exact identical MHC class 1 genes on the surface of their cells. So these are important for self-identification. So when your immune system is wandering around your body, these immune cells are looking for invaders. It basically can ask your cells for ID, and if the cell can present an MHC class 1 that says that it belongs here, then it won't be destroyed by your immune system. Right? So these are glycoproteins found on the surface of all nucleated cells. Now, MHC class 2 proteins, these are more specific. So we don't find these on all cells. In fact, we only find them on cells that we refer to as antigen-presenting cells. So those are dendritic cells, macrophages, and B cells. Antigen-presenting cells, their whole job is to find antigens and then present them to T cells. And MHC class 2 proteins are the molecule that they use to do that presentation. right? So they basically, this MHC class II protein we find on the surface of those APCs, and that's going to essentially hold an antigen and bring it to a T cell, right? And then MHC class III proteins are genes code for complement proteins. And remember, the complement proteins are those defense proteins that are just floating around in your bloodstream, waiting to be told to attack a pathogen. All right, so let's take a look at how your innate immune response interacts with your adaptive immune response. Right, so both of these responses work together. You actually need your innate immune system to help get your adaptive immune system going. And it works through those MHC class II proteins. So for example, here in picture A on the left side of your screen, we see a phagocyte. That phagocyte is, probably a macrophage or a dendritic cell. So he's capable of eating bacteria through endocytosis. So he's going to bring in that bacterium, he's going to digest it, and then he's going to find part of that bacterium, just part of it usually, and present that part of it as an antigen on the surface of his cell. So once he does that, then he becomes an antigen presenting cell, and he's capable of taking that antigen to a T cell to start the adaptive immune response. All right, so those antigen-presenting cells, and mostly dendritic cells and macrophages, but B cells can act as antigen-presenting cells too. And again, these cells are capable of phagocytosis. They will eat pathogens, digest them, and then present just an antigenic part of that pathogen with their MHC class II proteins. All right, so before we go any further, I want to just describe the outside surface receptors that we find on B cells and T cells, because again, your adaptive immune response is heavily based on antigens and also cell surface receptors. Right, so if I'm looking at a B cell here, so on the left side of your screen, we have a B cell. A couple things to note about a B cell. One, we've got this cell surface receptor here, and that's an MHC class II. So that's what an MHC class II surface protein looks like. So that is where antigens can be presented. On the surface of B cells, we also have immunoglobulins. Immunoglobulins are these Y-shaped molecules that we find on the surface. And then this B cell, although we don't see it in this picture, would also have an MHC class I, some sort of like glycoprotein flag that sits out here and says, I'm part of this body, I belong here, don't eat me, essentially. So I'll put Al for me. And then T cell over here on the right-hand side, he's got some different markers. So first thing we notice is he does not have any MHC class II. He will also have an MHC class I, but he doesn't have any MHC class II. He doesn't have any immunoglobulins, but he does have is this T cell receptor, also sometimes called an antigen binding site. And that T cell receptor is where the T cell will receive antigens. So when they are presented to him, he can receive them at this antigen binding site. Now, on either side of the antigen binding site, we see some other surface proteins here, and those are CD3 markers here, and then this larger CD4, the brown one in this picture here. So these are common for helper T cells to have this CD3 and CD4. And they basically act as guides. So they're going to help guide the presentation of an antigen. All right, so keep in mind, this is what the surfaces of both of these cell types look like. All right, now let's look at what that presentation of antigens looks like up close. All right, so in this picture up here, we've got an antigen presenting cell. So this looks like a dendritic cell to me. He's capable of phagocytosis, so he's going to bring in a bacteria through endocytosis, combine that phagosome with a lysosome, digest that bacteria into multiple different little pieces, take some of those pieces here, and then present them on the surface through his MHC class II. So it takes a little piece of the bacteria, presents it on that MHC class II on the surface of the cell. All right, so now he's an antigen presenting cell. He's got this antigen that's a determinant from that particular bacteria. So whatever antigen that we see here is going to be specific to whatever microbe it is he ate, and it's not going to be recognized by any other or recognized as being any other type of microbe. So this antigen presenting cell then is going to take this down to a T cell down here, and then it's going to interact with the T cell receptor. So this is a good picture here where we can see the MHC class II of the antigen presenting cell presenting that antigen so that it is received by the T cell receptor, and the CD3 and CD4 molecules are there kind of holding all this interaction in place so that we can have recognition by that T helper cell. Right, so the T helper cell at this point now has received the presentation of the antigen. He knows what bacteria is invading the body. Right, it's basically like you can think of like your dendritic cells as or macrophages or whatever antigen presenting cells as kind of like foot soldiers in this war on pathogens. So they're going out, they find the enemy, and then they bring part of that enemy to the T cell, and the T cell is going to be kind of like the general who's in charge of all your armed forces here. He's going to be the one that's then going to direct the rest of the adaptive immune response going forward. Right, so now this is basically the handoff between your second line of defense and your third line of defense. So this phagocytosis that's happening as part of your second line of defense is going to bring the recognition of this pathogen to the T cells, which are part of your third line of defense or your adaptive immune system. Right. So again, antigen presentation, incredibly important process for your adaptive immune system. So if we've got an antigen being presented to a T cell, usually that T cell before it's being presented is going to be considered inactive. So it's an inactive helper T cell. It's not doing anything yet. He's just sort of waiting around to be activated. Once we present the antigen, then he becomes an active T helper cell. Right. So here's another kind of picture of that antigen presentation. This is a typo. It should say MHC class II is presenting that antigen to the T cell receptor. But then once that has happened, now this active T helper cell is going to be able to activate a whole bunch of other T cells. So he's going to activate some cytotoxic T cells, also called CD8 cells, because they have cell surface receptors that are CD8. Those CD8 cells are going to be capable of going out and destroying cells that are foreign invaders, and they can destroy cells that have been infected in your own body, similar to what a natural killer cell does, except this is a much more specific process. And that helper T cell can also activate memory T cells. So memory T cells are going to remember this particular pathogen for decades to come. Right? So these are cells that, well, if you're lucky, these are cells that are going to hold a memory of that particular antigen. So if the body ever encounters that same antigen again, now you have memory cells that are built up and ready to defend you against that particular antigen. All right, so how does cytotoxic T cells work? They work in there are a couple different mechanisms of action here. So we mentioned the granzymes and the perforins. So granzymes are proteins that can be released by a cytotoxic T cell. They're going to trigger that cytotoxic T cell, or I'm sorry, trigger that infected body cell to undergo apoptosis, which is that programmed cell death. So the granzymes cause that cell to destroy itself. And then whatever microbes were inside that guy, usually viruses or intracellular bacteria, are then going to be released. And usually there's a waiting phagocyte nearby to ingest those microbes. Some cytotoxic T cells also have chemicals called granulysins and perforins. So perforins do what they sound like. They perforate, so they make a little hole in the surface of an infected body cell. And that's going to allow the granulysins to enter, and those granulysins will destroy the microbes from within. Right, so in either one of these situations, the infected body cell will die, but we prevent the future spread of those microbes that were invading it. Okay, all right, so this adaptive immune response We've been mostly talking about the T cell line. The T cell line is called your cell-mediated immune system because we're directly attacking pathogens or directly destroying your own body cells. So the T cell line is the cell-mediated line, directly goes for those cells. All right, so that's where we're producing cytotoxic T cells. We can produce memory T cells, regulatory T cells that are going to be important to maintain cell-mediated immunity. Another thing that your T helper cells can do is initiate the other wing of your adaptive immune response, which is the antibody-mediated response. So antibody-mediated response, this one involves the B cells, so your B lymphocytes, which are capable of transforming into plasma cells, which make a whole ton of antibodies. Right, so antibody-mediated immune system is sometimes also called your humoral immune system because instead of this wing of your immune system going directly after cells, we release antibodies into the quote-unquote humors of your body or the fluids of your bodies. So that's why it's sometimes called the humoral immune system. Okay, so here's how these two branches of the innate immune system work together. Right, so remember, we get an infection from an initial, whatever it might be, bacteria, virus. It's going to have to get past your first line of defense first. So it's going to have to get through some body surface, going to get into the body and trigger the second line of defense. At that point, a phagocytic cell will ingest it and then present antigens on its surface here to a helper T cell. Then the helper T cell, like I said, is kind of the general in charge of this whole response. He's going to initiate a cell-mediated response and an antibody-mediated response. So he's going to trigger other T cells to go do their thing. So trigger cytotoxic T cells and T memory cells. But he's also going to trigger B lymphocytes to go do their thing, which we'll talk about in a second, how they go about making antibodies. All right, so here's what B cell activation looks like. So B cells can be activated directly by a microbe, but most times they're going to be activated from a T helper cell. So a T helper cell is going to trigger a B cell to become activated. When a B cell becomes activated, it's going to do a similar thing to what the T cells did. So T cells kind of clone themselves and differentiate and make all these different types of T cells. B cells are going to do the same thing. They're going to make different types of B cells. One of the most prominent types of B cells to make are plasma cells. And plasma cells are cells that are short-lived, right? They don't live too long. And their whole job is to produce antibodies. So they're basically little tiny antibody factories. And they're going to be making antibodies that are specific to whatever microbe they were activated to fight. Right, so the antibodies that these plasma cells are making are specific to this microbe up here and this one alone. They're not going to be responsible for attacking any other type of microbe. And then we can also develop memory B cells, and memory B cells are going to remember this particular pathogen for future infection. All right, so there's that activation again. So we activate, we develop the B cell clones. Those B cell clones are going to become plasma cells that make antibodies, or memory B cells that are going to be long-lived. And we make sure that those clones are directed towards that particular microbe and no other microbes, and certainly no other body proteins. All right, so what's the point of developing all of these antibodies? Right, so what do antibodies actually do? They don't directly kill anything, right? So these are not like cytotoxic T cells. These are proteins. But the proteins have 5 major different functions in aiding your immune response. So one thing that they can do is neutralize. So they can basically surround toxins that have been created by some bacteria. And once they surround those toxins, they essentially neutralize them. They can immobilize pathogens. So a lot of pathogenic bacteria are capable of movement, but if we completely surround them with antibodies, then they're no longer capable of moving so that they can be spotted by a phagocyte of some sort, they can agglutinate them. So agglutination is connecting multiple different pathogenic cells together, so that they're kind of all locked with one another and they're not able to swim independently. Antibodies are also important for activating the complement system, which are those little defense proteins that you find in your bloodstream, and then also enhancing phagocytosis. So basically acting as a trigger for macrophages to come along and eat a pathogen. All right. So in order to make sure that we're doing this right, that your immune system is developing the clones necessary to attack a particular antigen, we have to look at the clones that we're making and make sure that we're picking the right one that fits that antigen, and we're not picking any that are going to attack self cells. Right. So this process is called clonal selection. So clonal selection is making sure that the clones that we're making are the ones that are going to attack the particular pathogen that we're dealing with, and hopefully not any of our own cells. All right, so here's what an antibody looks like. So in this one in particular here is an IgG. So an antibody is a protein. There's some basic parts to this protein that you should be familiar with. One, it's comprised of two heavy chains. I'm going to kind of highlight the heavy chains here. So these are these long chains that are kind of this Y shape. And then two light chains. So I'll highlight those as well. The light chains are kind of on the outside of the arms of the heavy chains. Now, the heavy chains are held together by disulfide bonds. So these two little sulfur bonds in the middle here is what's holding the two heavy chains together. The stem region, or this kind of bottom region, this is sometimes referred to as the constant region, because this part of an antibody is going to be look the same across no matter what antibody we're looking at here. The region up here, these are called hypervariable regions. This is what is going to make an antibody so very specific. So we can develop antibodies that are going to be able to recognize specific molecular patterns on the surface of an E. coli, for example. And the same molecular pattern that they recognize on that E. coli is not going to match with anything else, any other pathogen on the planet, right? So these guys are capable of incredibly specific shape recognition, and it's because of these hypervariable antigen binding sites on the ends of the arms of that Y-shaped molecule. All right. So again, the roles of antibodies are the basic roles are neutralization, agglutination, opsonization, complement fixation, and then opsonization. Did I say that one already? Take a picture here. Okay, so here's a really good picture of those different functions of the antibody. So right here we've got opsonization. So in this picture, we've got a bacterium. It looks like a rod-shaped bacterium that's totally surrounded by antibodies. So both hypervariable regions of each Y-shaped antibody are attached to surface proteins on this particular pathogen. So he is considered opsonized now. That opsonization is basically going to flag this guy as being bad, so that this nearby macrophage can come by and eat him more quickly. So he eats them, antibodies and all. Right? So the antibodies are sort of like a flag to say, here's a bad guy, come and eat him. So that's opsonization. If we do that same thing with a virus, so we're completely surrounding the virus here with antibodies, those antibodies are covering the spikes of the virus. And remember, spikes of a virus are what allows a virus entry into a host cell. And if a virus can't get into a host cell, it can't do anything. So by surrounding the viral spikes, we basically neutralize that virus. Down here, this is what agglutination looks like. So agglutination is when each arm of that Y-shaped molecule is attached to a different pathogen. And that's going to help the pathogens to clump all up together. And once they're clumped up together, they're basically sitting ducks for for a phagocyte to come and eat them. The next one here is complement fixation. So where an antibody has linked onto a pathogen, it will trigger activation of the complement system. And remember, complement are dissolved proteins in the blood that are capable of poking holes in the surface of pathogens. So that's going to help with the bacterial cell lysis, destruction. And then the last one represented here is antitoxicity. And so that means if a bacteria makes a particular toxin, we can also surround that toxin with antibodies and essentially neutralize the toxin. Right, so in each one of these scenarios, the antibody itself isn't destroying the microbe, but it is helping the rest of your immune system do so in a really specific way. Right, so these antibodies are going to be very specific and help your immune system trigger the destruction of those pathogens. All right. There are 5 different major classes of antibodies. So the classes of antibodies are IgG, IgM, IgA, IgE, and IgD. So that stands for immunoglobulin G, immunoglobulin M, and so on. The most common one, the one that you have the most of in your body at any given time, is IgG. So about 80% of your antibodies at any given time are going to be IgG. These are very protective against microbial infections. This is also the only class of antibodies that's capable of crossing the placenta to help protect the fetus. The next class here is IgM, immunoglobulin M. These ones are usually the first ones that your plasma cells make in response to a new infection. So you get a new cold that you've never had before. You're in the process of the B cell cloning and clonal selection, we're making these new plasma cells. Plasma cells are then going to start producing new antibodies against that pathogen. IgM is the first one that they're going to be producing, and then they'll follow that up with a lot of IgG afterwards. IgA is an antibody that we find in mucous membranes. We find it a lot in bodily secretion. So you produce IgA, gets released in your sweat, in your tears, in your saliva, in breast milk. We find it all along the mucous membranes of your GI tract and respiratory tract. So that's kind of like a protective coating for areas that kind of interact with the outside world. Immunoglobulin E, IgE, is involved in allergic reactions and parasitic infections. IgD, we don't really know what IgD does. So it's definitely an antibody that we find on the surface of B cells, but we still don't quite know what its function is. All right, so here's what these guys look like. So again, IgG, that's your most common one. That's going to be about 80% of your cells. Or 80% of your antibodies at any given time is IgG. It does cross the placenta. It exists as a monomer, meaning just the one single Y-shaped molecule. IgA can exist as a monomer or a dimer, meaning 2 of them together, like what's going on here. And this one is found associated with mucous membranes released in the milk and things like that. IgM, that's your kind of first responder antibody. So this is the first one that's produced by your plasma cells after an initial infection. He usually exists in this pentamer form, so 5 of them kind of attached together. IgD, again, we don't really know, but it usually occurs as a monomer. And then IgE also occurs as a monomer, and that one's going to be associated with allergic reactions and parasitic infections. Okay, so here's one of the cool things that your body does with this adaptive immune response and why it's so important and why it's so much more special than that innate response. And that is the production of antibodies over time. So looking at this chart down here, so say we get a cold, all right? So your first exposure to this cold, your immune system will slowly start producing B cell clones that are capable of recognizing this particular cold virus. Those those plasma B cell clones are going to start to produce IgM, immediately, right? So that, well, as soon as they possibly can, they'll start producing IgM. So we get this kind of wave of IgM, and then that wave kind of dies down after 2 weeks. Somewhere in there, you start producing IgG as well, and the IgG levels will kind of stay a little bit higher for a little bit longer. Then, so you recover, right? You recover from the cold. Your immune system fights it back with the use of all of these different antibodies, and you feel better. If then someone reinfects you, so you encounter that same cold virus again, someone sneezes on you, whatever, you're exposed again. The second exposure then is going to look a lot different than the primary exposure. So second exposure, yeah, we're going to be making that IgM, right? Roughly the same amount that we made during the first exposure. The major difference here is this incredible spike in IgG production. So you get this huge spike in IgG, and that's basically going to wipe out that cold virus or whatever infection we're talking about here immediately or really, really quickly to the point where you're not even going to feel sick. So you probably don't even know you've been reinfected because your immune system basically wiped out the infection before any damage could have been done to your body. Right, so this is why your adaptive immune system is so special, right? So that you're basically only catching the same diseases once, and then if you encounter them again, your body is prepared to fight them off. This is also the same mechanism by which vaccines work. In a vaccine, you basically the primary response is the vaccine itself. So you're injecting yourself, or a medical professional is injecting you with antigens, and your body is then making antibodies as a result that are going to be able to fight those particular antigens if you actually encounter them in the real world. And this is also why sometimes after getting a vaccine, you can feel a little bit under the weather. And that's because a lot of your feelings when you feel sick is that it's exhausting to build this immune response. It takes a lot of energy. So after a vaccine, you may feel a little bit tired, a little bit under the weather for a couple of days as your immune system responds and builds that memory response for the next infection. Okay, so the ways that we acquire adaptive immunity, there are a few different ways here. So first way we'll talk about is natural active. So natural active acquisition of immunity means, natural means you encounter this pathogen in a normal way, right? So like this little kiddo here has chickenpox. He probably got the chickenpox by interacting with another kid that had the chickenpox. So that's a natural way to acquire the disease. And once he's acquired that disease, he actively is developing his own antibodies, right? So he's building an immune response against this naturally acquired pathogen, right? So that's natural active. What's happening in this picture over here is the woman is nursing. This is natural passive immunity. So natural because nursing is a natural process. Passive because the baby is passively receiving IgA through the milk. So the baby is getting IgA, he's getting antibodies, but he's not making his own antibodies. So that's passive. So active if you're actively making them, passive if you're getting them from somebody else. And both of these situations are considered natural. There are also artificial ways to acquire immunity. So artificial active immunity is vaccines, basically. So artificial because you've been injected artificially with these antigens instead of acquiring them naturally. And active because once you've acquired those antigens, then you start developing the antibodies to fight it. So that's artificial and active because your body's still doing the work to make the antibodies. Artificial passive would be like the passive injection of antibodies. So like anti-venom medications are often considered this because one, you have to go to a doctor to get it injected to you. So that's artificial by its nature. And passive because they're basically giving you those antibodies instead of you developing them on your own. All right, so these are both artificial and they can be either active or passive. All right, so some questions here to wrap it up. What is an antigen? What are antigen-presenting cells? What cells do antigen-presenting cells present to? And then what kind of white blood cell is a T helper cell? So here's your answers. So an antigen is any molecule that is recognized by the immune system as being foreign, right? So they often tend to be larger, more complex organic molecules, but anything that your immune system recognizes is going to be antigenic. The antigen presenting cells that you have are macrophages, dendritic cells, and B cells. So those are all cells that are capable of presenting antigens through their MHC class II surface receptors. Antigen-presenting cells present their antigens to T helper cells, and T helper cells are lymphocytes. Right? Which cells produce antibodies? Which cells kill virally infected cells? What antibodies do we find in secretions? And what antibodies are involved in allergic reactions? Right, so there's your answers. Plasma cells create antibodies, B cells create antibodies. The cells that kill virally infected cells are cytotoxic T cells, if it's a specific immune response, or natural killer cells can do this non-specifically. What antibodies are found in secretions? That's IgA, and the antibodies involved in allergy is IgE. All right, last set of questions. What's an antibody titer? What is opsonization? What does opsonization help with non-specifically? And then which is the only class of antibodies that crosses the placenta? Right, so the antibody titer is just the measurement of the quantity of antibodies in serum at any given time. So it basically lets you know how immune you are to a particular infection. Opsonization is the surrounding of an antigen by multiple antibodies. So just multiple antibodies grabbing onto surface receptors and completely surrounding a pathogen. Opsonization helps with phagocytosis, right? So it triggers a macrophage to come by and eat that pathogen. And the only class of antibodies that crosses the placenta is IgG. All right, so I know this was a long one. The immune system's a little bit complicated, but I hope this helps. Let me know if you have any questions.